It's a little bit of a solemn day. I was in New York City this morning and saw a bunch of firemen in their uniforms, and it's a good reminder. I mean, it's one of those days, at least for those of us that are a little older, where in all likelihood, all of us remember that day very well. I love reading. It's one of my primary passions in life, I've come to realize, and I've written a number of reviews of books, including the Feynman Lectures and also What is Life? And one of the things that came up in writing my review of the 75th anniversary of What is Life is the fierce, intense rejection of Schrodinger's lectures and book by both Linus Pauling and Max Perutz. And I thought this was kind of a funny remark from Pauling, who said Schrodinger's discussion of thermodynamics is vague and superficial to an extent that should not be tolerated even in a popular lecture. And I fiercely disagree, actually, with both Pauling and with Max Perutz. I think they were obnoxious. And I think that Schrodinger he knew his thermodynamics very well, but it does prove what a horrible thing it is to have to give a public lecture. So I hope that you'll bear with me. So here's the outline. So what I want to do is I want to basically tell you three things. And maybe I've aimed too low, but the hope is that you won't need to pull out your iPads or your calculation pad and start doing calculations, that I'll hold your attention. So I'm going to start out, and it's probably going to take me 20 minutes to introduce what I even mean by this notion of the genomic Rosetta Stone. And then once that's in hand, what I'll do is tell you the main technical part of my talk, although it will likely not be that technical. And that has to do with trying to figure out how to decipher genomic hieroglyphics. And I hope you'll understand very well both what the question is and how we've attacked it. And then the last part will be much more brief, but in a way is the actual goal of the work, and that is to be able to write what I will call genomic poetry. And you'll see what I mean by that as I go along. So first part, 20 minutes probably, and trying to give you a sense of what I mean by the genomic Rosetta Stone. So collections of letters are wild, and I think it's fun to think of a collection of letters as a vector. So if somebody writes a book, then I can think of that as a 10 to the 6th dimensional vector. And maybe there are people in this room that can read this amazing, beautiful, wonderful paragraph, but I'm not one of them. And so that leads to questions about what do letters mean. And this one in particular is this fantastic final paragraph of On the Origin of Species. There's grandeur in this view of life, with its several powers having been originally breathed into a few forms or into one, and that whilst this planet has gone cycling on according to the fixed law of gravity, from so simple a beginning, endless forms, most beautiful and most wonderful, have been and are being evolved. Beautiful letters strung together in a way that both conjures meaning and also emotion and insight. So books, in a way, are our collective memory. And here I show you 4 collections of letters that I'm particularly fond of. So this is from book 1, Proposition 47 of Euclid, the Pythagorean theorem. That is Isaac Newton in the Principia, showing us by essentially trapping the area under the curve between the circumscribed and the inscribed set of rectangles, and then setting the width of these rectangles ever smaller, that he can compute the area of such a curve, what we now know of as the integral calculus. A lot of what I'm going to say today is going to at least touch on whales over and over again. So that's Moby Dick. And then finally, these are the nascent notes in which Darwin started to think about phylogenetic trees. Again, probably many of you know this, but this is from his Transmutation Notebook B. And I love this. He says, "I think," and then he draws this little tree. So there's basically a giant encyclopedia which is written in a different language, and that's the Genomic Encyclopedia. And that encyclopedia is written in a language with 4 letters, A's, C's, G's, and T's. And in a way, what I want to do is I want to talk about the meaning that lurks inside of those letters. But before doing so, I want to set up, in a way, what I hope is something that's going to bother you. So to do so, I want to evaluate 4 different collections of letters and evaluate how big they are and how meaningful they may or may not be. So I asked ChatGPT today, I already knew the answer, but I asked it how many letters are there associated with Wikipedia in English. And interestingly, it gave me 5 times 10 to the 10th letters. And just to give you a sense of that number of letters, I wanted to tell you about a beautiful, fascinating metaphor. So I want you to imagine that the diameter of DNA is like this. In other words, I scale up DNA so that that's the diameter. Then the machinery that copies DNA, the replisome, is the size of a FedEx truck. It's traveling at roughly the speed of a jet aircraft. It's delivering on both sides of the street a package every 10 centimeters. Its daily journey takes 40 minutes, and once every 3 years, it makes one mistake. So that's the fidelity of DNA copying. I can tell you, this sounds pretentious, but as somebody who has written some books, it's really something that pisses me off when I see Kindle books, because they have, I would say, an error per page, roughly speaking. Quite different from the fidelity of copying of DNA. If we go to the Library of Congress, what we would find is on the order of something like 30 to 50 million books, depending on how you count it. And if I think of there being something like a few, I'm going to use F for few. So few times few is 10. So if I take few times 10 to the 7th books times few times 100 pages per book, few times 100 words per page, and let's be generous and call it 10 letters per word, that's going to lead to around F times 10 to the 13th letters. What about ChatGPT? So it's a little harder to figure out how many letters were actually used to train ChatGPT. If you ask ChatGPT, it will be a little cagey about that question. But I think that a rough estimate is something like 6 times 10 to the 11th letters. And I find it kind of crazy and remarkable. I did this just the other day, make a beautiful, so naturalists these days often use satellites. Instead of going out on sailboats and landing and hardship, they use satellites to examine the world around them. And so I said, make a beautiful image of Charles Darwin riding a satellite looking down on planet Earth, and that's what it produced for me. It says, here's the image of Darwin riding a satellite while looking down on planet Earth. It captures a whimsical view of Darwin in space. Well, and then the last example, and the one that bothers me, is this is a measure from the sequence read archive that tells you that we have accumulated 10 to the 17th A's, C's, G's, and T's in the sequence read archive. Why do I make this point? Because I want to contrast it with those other 3 examples. You might, I mean, if you were really demanding, you would say, well, then that must mean it's like 10,000-fold more information, or 10,000-fold more enlightening, or something like that. I'm willing to be challenged, but I'm personally telling you that I'm not persuaded that we have that kind of understanding. So within every one of our few times 10 to the 13th cells, with the exception of red blood cells, we have roughly 2 meters worth of DNA. Just to put that in perspective, the distance between bases is 1/3 of a nanometer. There are roughly few times 10 to the 9th base pairs in the human genome. So few times 10 to the 9th times 1/3 gives you 10 to the 9th nanometers, which is a meter. And since you have 2 copies, that's how much DNA is in each one of our cells. And just to give you a sense of that, hopefully this will blow your mind. I always just find it kind of astonishing. If I take my 5 terabyte hard drive, which I have with me, and I examine how many letters are saved in that, it's roughly 5 times 10 to the 12th, and it's in a volume of about 100 cubic centimeters. If you consider a virus, such as I chose influenza, which is super interesting, beautiful virus, just an obnoxious side comment, when people were talking about SARS-CoV-2 and influenza being the same, I was offended because scientifically they're so wildly different. This has 8 separate RNA molecules as opposed to 1 long molecule. But at any rate, look at the information density. 10,000 letters per 10 to the 5th nanometers cubed, which is a density of roughly 10 to the 20th letters per cubic centimeter. So I had a fantasy. So this morning, I had my friend Nigel actually draw this up for me. So this is taking the genetic code in order to make a library of all the books ever written. So what you would do is you choose a particular configuration of A's, T's, C's, and G's in order to highlight a particular letter. So for example, TTT will map to the letter A, and let's see, I don't know, so G will map to the Greek letter rho, and so on. And what I think is amusing about that is that a little tiny test tube of 5 cubic centimeters will have more than enough bacteria to carry extra letters so that I can put every book ever written. Okay, so these molecules also are documents of evolutionary history, as was noted by Zucker, Kandel, and Pauling, as I show you here. I love the title of this paper. And in it, they make this very provocative, beautiful remark. In macromolecules of these types, there is more history in the making and more history preserved than at any other single level of biological integration. So let me try to tell you a little bit about that. So if I think of many, many generations ago, some template DNA, the way I want you to imagine this is that when the DNA is copied, every base flips a coin. And the coin is extremely dishonest. The probability of the heads is 10 to the minus 10th. And so you rarely, but now and then, get a mutation. And that mutation, like for example here, G got converted into A. And if we do this over millions of years, mutations accumulate. And so the accumulation of mutations is a stopwatch. And that has been used to great advantage in order to think about molecules such as hemoglobin. And we can use the DNA sequence or the protein sequence of hemoglobin to measure the relatedness of organisms. For example, my hemoglobin has 142 amino acids, and it's identical, as is yours, to that of a chimp. For a gorilla, you'd have one difference. Compared to a cow or a horse, you'd have more differences. And compared to a fish like carp, you'd have even more differences. So just as a little aside, maybe you've never thought about this before, but do whales have belly buttons? How many of you think they have belly buttons? OK, that's good. So yes, and the reason I say that is that they're placental mammals. And so I want to talk just for a sec about how this DNA sequence business could be used to learn a little bit about the history of life on Earth. So this is a baleen whale. They basically, every day, take on, every single lunge they make, they dive, let's say, between 100 and 1,000 meters. They'll make 10 lunges per dive. They will go exactly to where they find krill. And every one of those, they're 30 tons, they'll take on between 5 and 10 tons of water with each and every such lunge. But they're toothless. So do they have the genes for enamel? They do. And there's a beautiful paper. This is, again, just to give you a little bit of a sense of this notion of stored history in genomes. So these are all sorts of different animals. You can tell we have toothless whales, anteaters, armadillos, sloths, et cetera. Some of them don't have teeth. Some have teeth without enamel. And DNA sequence can be used as a window onto the differences between these animals. And the thing that's very funny about the genes in whales, for example, is that they're sort of littered with what are called stop codons, meaning the genes are no longer functional. They're fossils. They're useful as fossils, but they're no longer functional. So back to this DNA language. I'm basically trying to set up this notion that this set of A's, C's, G's, and T's is very interesting. It holds a lot of keys to the history of life on Earth. It tells us about the function of organisms. But there's a dark secret, which is what I'm headed for. So there's a beautiful poetry. Already in the 1960s, especially Marshall Nirenberg did these quite amazing experiments that allowed us to understand the nature of the genetic code. And the secret, or to my mind, the dirty secret, is that there's a huge fraction of the genome which we still do not understand. And I'm going to focus on the regulatory region. And the reason I post this up here is that that particular set of bases, I also ran that by ChatGPT today just to see what it would say. It was one of its worst performances ever. So, so this set of bases comes from E. coli, one of the best understood organisms on the planet. And it's just a part of its regulatory apparatus. And we just basically don't know what it does. So I've always been amused by what happened at the end of the 19th century in physics. And one of the things that happened is Kelvin wrote this paper. Probably many of you have heard of it, 19th century clouds over the dynamical theory of heat and light. And I think this current article by Art Lander is really clever, just the abstract alone, and I'm going to read it to you. A culture's icons are a window onto its soul. Few would disagree that in the culture of molecular biology that dominated much of the life sciences for the last third of the 20th century, the dominant icon was the double helix. And to my mind, the double helix, the Keeling curve, the symbol for radioactivity, these are truly moving images. And the DNA double helix is a beautiful and moving image. In the present postmodern systems biology, however, it is arguably the hairball. And I guess what I would say, we don't all have to agree, but I personally find that to be incredibly ugly. And I wish we could do better. And I guess in the end, often my metric is, what am I going to do when I'm faced with a bunch of 18-year-old haters that are forced to take biology at Caltech? How do I deal with these people? And I don't want to ruin their day. And showing them hairballs, I think, is part of the bad reputation. So what I'm going to try to do is demonstrate this idea that we have basically a Rosetta Stone problem. So this is just to note that both Thomas Young and Champollion, they spent a lot of effort in trying to understand what was going on with the Rosetta Stone. And to get to the point where you'll understand what I mean by a corresponding problem in the context of genomes, I need to tell you a little bit about the central dogma. So the notion is there's a journey from DNA to mRNA to protein. There's a little molecular Xerox machine called RNA polymerase that copies the message in the DNA and turns it into this molecule of mRNA. And then there's a universal translating machine that speaks two languages. It speaks the nucleic acid language and the amino acid language and turns the information in the mRNA into a sequence of amino acids in the form of a protein. But there's a set of interventions shown over here. And what I mean by that, whenever I think of this, I always think of New York City. So there's fancy clubs in this town. I wouldn't frequent them, but I know there are such things. And there are people standing at the doors, right? And they decide who gets to come in and who doesn't. And so those essentially are activators and repressors. So the language I'm going to use is that there's a kind of molecules known as repressors that bind to the DNA and close a gene for business. And then there are others known as activators that bind to the DNA and they actually enhance the probability of this little molecular Xerox machine from being there. And so here is the dark secret, in a sense. And that is that we do not have a systematic way of examining the part of the DNA whose job it is to control when and where genes are deployed in general. And I will try to hang some meat on that. So that's the first part of what I wanted to say. So just trying to make the argument that there's something more for us to figure out about the properties of DNA. So basically my talk is about a puzzle. And the puzzle is very simple. It is, what is the meaning of these letters? One night, Caltech students they're a very impressive bunch, and they're impressionable. And at 11 p.m., when I was teaching freshman biology one night, I sent them, the whole class, a sequence like this with no explanation. I just simply sent it. And it was really funny because the next day nobody got any sleep and they were all trying to do these, all this code cracking and all this stuff. And really all it was just like a chunk of the E. coli genome, which is standard stuff that we still don't understand. So let me really try to make the point. So this is an attempt to use careful analysis to tell you how bad the problem is. So let's start with the upper case. So this is a cartoon of E. coli. It has a genome of 4.6 megabases, so that means roughly 4.5 million bases in its genome. That's one complete works of Shakespeare. And what I'm showing you up here in red is the fraction of genes for which we don't know anything about how they're regulated. And when I say regulated, again, the rules for when the genes are deployed in space and time. In the case of other famous organisms like Drosophila, C. elegans, yeast, the situation is far, far worse. And if I were to actually ask about an organism that I care about a lot, such as whales, or since the dominant biomass of mammals on planet Earth is cows, like cows, I would know roughly nothing. So maybe some of you have seen these kind of suggestive diagrams before. They're meant to elicit a reaction on your part. They're meant to suggest the idea of a wiring diagram. And so the one on the left is somewhat of a statement about the anterior-posterior patterning in a fly embryo. This is the work of many years of Eric Davidson, in which he was thinking about endomesoderm in the context of the sea urchin. And there's many issues about this that we could critique, and it's fun to think about the shortcomings even of these diagrams. But an even more honest statement is just, this is the exception, not the rule. For almost everything you might care about, we don't know what the wiring diagram is. So if you were to take some sort of a freshman or high school biology class, these are the kind of cartoons that would be shown to you as people explained gene regulation and made it sound like we've got it nailed. That's reality. Not that, that. Okay, so now we hit the year 2010, and I do have permission from Kurt Cowan to show this. So probably many of you know that in the context of the journal PNAS, people that are members of the National Academy of Sciences have the right to send out a paper to their colleagues asking for a comment on their work. And Kurt sent me this paper, which was written by, in particular, his grad student, Justin Kinney. And I was in Kauai at the time. And I was so blown away by this that I called these guys and I said, let's have a conversation tomorrow morning when I'm driving to Honolulu, because I'm going to be going surfing. The time is perfect for us to have a chat. And we have now been on, that's 2010. We are now on our 14-year journey. We've written 2 papers based on this, but it has been really the primary driver of what we've been up to. And so I'm going to try and explain to you what the idea was. And to do so, we'll use Anna Karenina. So at the beginning of Anna Karenina, probably many of you know, there's this famous statement. The translations are different that I used. All happy families resemble one another. Every unhappy family is unhappy after its own fashion. So again, I'm kind of on a rampage with ChatGPT. There was an article in the New York Times, or an opinion piece a few weeks ago, I don't know how many of you saw it, about ChatGPT in the context of the classroom. And I've been doing experiments ever since, just trying to see what happens if I try to get in total student mentality and use it with abandon. So I asked it to write a Python code to take this sentence and to mutate it at the 10% level. I also had it do it for other mutation rates. And the kind of stuff that comes out of it is, like you can see, the word way became ivy. The word way became y. Over here was a hippie one of the times, and so on and so forth. So the concept is the following. What we're going to do, I will show you in the context of a cell, we're going to take some region of DNA that drives a gene called a promoter. We're going to mutate it at the 10% level. And then we're going to ask the cell, how much did you care that we mutated the DNA? And the way we'll know that it cared is by how many mRNA copies that it makes. So in other words, some letters matter and some don't. Let's take the word walk. If I mutate the letter A, it has very little information content. Why do I say that? Because you'll still know it's the word walk. I can't think of another word. Maybe some of you will, but I don't think there's another word that it's legal for me to substitute A with another letter and get a valid English word. On the other hand, if I mutate the K, for example, to a G, you will not know whether I started with walk or wall, right? So the L at the end of the word walk, or the K, is more meaningful than the A is. And so what we're doing is we're trying to read out which bases matter for the amount of gene expression. So the readout of gene expression is how many mRNAs there are. The way we know which promoter we had is we have a barcode that allows us to tell what the mutation was. And the result of that is we actually, the data, if you want to think of it that way, is counts, a mapping between counts and mutations. And that's going to be the basis of this detective story of deciphering the genomic Rosetta Stone. So again, to come back to Anna Karenina, we mutate, and then we ask the question, what letters mattered for understanding? Clearly, this W and that A are quite meaningful, because if I get a double mutation to I and V, this becomes the word ivy. I'm not allowed, by the way, to use context of the neighboring words, although if we're smart, you might be able to do that too. OK. So now I'm going to tell you a bit about the protocol. I'm going to just go through each step of this, so you don't need to understand it just yet. But it's basically to go from complete ignorance, to being able to design with the result. In other words, I want to be able to engineer the gene expression at will by tuning sequence. I want to go from ignorance to that engineering option all in one fell swoop. OK, so how does this go? Point one, we choose which gene we're going to look at. We mutate it. We make a library of thousands of copies, each of which is mutated at some level. And that level matters. And there's a reason to choose different levels of mutation. And I'd be very happy if you asked me about it. The next thing we do is we install one of these per cell. And we make a huge library of them. And then we grow them in all sorts of different conditions. In our case, it's 40. Why do we do that? Because we have to try and find a way to perturb the cell in a way that it cares about. Not all genes care about all perturbations. For a lot of perturbations, a given gene is indifferent. It simply does not care. And there are genes for which we do not know how to get them excited. And nobody does. I mean, I find that kind of stunning. So that's what we do. And then what we do is we sequence. So what we do is we sequence how many counts there were. And we basically know the sequence of the promoter. And then we do an information theory calculation. And what's cool about that is that we're calculating the mutual information between which base is at site I and which bin of expression. In other words, do I care that you mutated this particular base? This is a mathematical representation of that degree of caring or not. So the result of that is a regulatory hypothesis. We have a cartoon, but we're not done. So that cartoon allows us actually to create what I'm going to call an energy matrix. This is a way to compute the binding of the transcription factor as a function of the particular sequence in KBT units. So it's like, it's real stat mech. And then finally, there's one missing point. Which repressor or activator binds that site? And to find that out, we use mass spec in a way that I'm going to show you in a little bit. And then when we're done, we have a new picture. OK, so let's talk about the first part about mutual information. So as many of you know, or perhaps all of you know, Shannon, to my mind, was like the Carnot of information. In other words, he said, I don't care if you use telegraphs or telephones or wireless or whatever. There are rules about information transfer. And one of his ideas was this idea of mutual information. So here's the paragraph I read earlier. And this is a table of frequencies of letters. I always find this amusing in the context of Scrabble. What are the scores associated with letters in different languages? Like, for example, the Q is not that virtuous in French, right? It's not the same virtue that it has in English, and so on. I can play this game. But this is not that interesting. What's much more interesting is this matrix. This matrix is, if my current letter is an N, what's the probability that the next letter is a G or whatever else? And so that's the mutual information. And the point is, if I know that table and you don't, and we decide to have a betting game, I will fleece you. I will definitely win, because there's a mutual information between the current letter and the next letter in a word in the English language. So the mutual information is a measure of the information when you know Y, what's the probability of X, essentially. And so here's what this looks like. This is in the case of a well-known gene. The details really don't matter. But basically what I'm telling you is that these are particular bases that matter a lot. The ones in between don't matter at all, really. And we can identify binding sites for repressors, for polymerase, for activators. All of these things emerge naturally without us doing much massaging. And then the second point, and this is really for me, the connection to statistical mechanics. I want to be able to calculate these things from first principles. I want to be handed a sequence and be able to compute the level of expression. So I can elaborate on that in a little while. Now, I've told you how we might make a hypothesis of where things bind, but we so far don't know what binds there. And this is really a needle in a haystack problem. So I don't know how many of you have seen this, but this artist has an exhibit or had an exhibit in Paris. You could go watch him in real time, but you could also watch on a webcam. So here's the way this goes. He basically takes a needle. He gets a bunch of hay. He pours it in there. He has a webcam, and then he finds the needle. So that's it. So let's do an estimate about that, and then connect to the biological setting. So we'll use Monet's haystacks as an example, and here's the way the estimate might go. So here's my haystack, and the volume of a piece of hay might be something like 10 centimeters by a quarter of centimeter squared, which is a few centimeters cubed per piece of hay. The volume of the haystack might be a few meters cubed. And so the number of pieces of hay in a haystack is of order a few million, a million to a few million. Why does that matter for the case of searching for proteins in the genome? Because what I'm showing you here, a very simple calculation to do, is how many proteins are in a cell. And there's a standard number that we can all carry around in our heads for cytoplasm. 1 cubic micron has a few times 10 to the 6 proteins. It's pretty straightforward to see that. If I take a 1 cubic micron bacterium, that's the same as a femtoliter, 10 to the minus 15 liters. So the mass is therefore a picogram, 10 to the minus 12 grams. A third of that is dry mass. So that's 30% of that picogram, and half of that is protein. So if you just plug in 30,000 hydrogen masses per protein, you'll find roughly a few times 10 to the 6 proteins. So that's this. That's the total number of proteins. And I just told you by pure thought. This is measurements from mass spec, but I just showed you how to actually estimate it so you can check whether you believe that this measurement makes sense. It's kind of depressing. I won't mention any names, but when there's a famous book that Jean Perrin wrote in 1910 called Atoms. And in it, it ends with 15 distinct independent measurements of Avogadro's number. And then there's a paragraph that says, so by the way, if you don't believe in atoms at this point, you're a lost cause for science. I mean, that's roughly what he had to say. So that's good. I like that. Here, when people try to count proteins, you can use fluorescence. You can use mass spec. You can do ribosomal profiling. There can be as much as 100-fold discrepancy. So it's important, at least to my mind, to ask, does the y-axis make sense? And so I just demonstrated to you that it does. This is the total number of transcription factors. There are of order 300 distinct types in E. coli. And this is one particular transcription factor. In other words, they come in at copy number 10. And that's roughly a needle in a haystack. So we need to find the one protein that binds our DNA, our putative binding site. And so how do you do that? You break open the cells like this. We have a magnetic bead with a little 50-mer attached to it that has our putative binding site. And basically, it's a fishing expedition. And what you're trying to do is get the thing you're hunting for to bind. We release it, put it in a mass spec, and we look for enrichment, like this. So now we've identified the transcription factor. And so my claim is that we have a procedure that takes us from knowing nothing to actually having a cartoon and being able to do StatMech. So back to my puzzle. So I started with this, and I asked, what is that sequence? What's it up to? And it's fun, because it's actually related to a very, very beautiful aspect of biology, which is lots of times there's feedback and control. One of the things that probably many of you learned is about AT and GC base pairing. Basically, there are certain conservation rules. And the cell knows about this. It knows very well that it wants to have the number of purines and pyrimides at the same level. And so it has a transcription factor that basically is keeping track. It's doing surveillance to find out, is there an imbalance? And if there is, fix it. And so maybe this is especially for the younger part of the crowd, and I don't know how all of you will feel about what I'm going to say, but I have very mixed feelings about scholarship. And what I mean by that is that I agree with this notion of yeah, that person doesn't know anything. But I also disagree with it, because lots of times those are the people who figure things out. So, like Schrodinger, there was another Austrian, super well-educated guy from Gymnasium that spoke 5 languages named Erwin Chargaff. He wrote a beautiful autobiography, which is called Heraclitean Fire. And he was pretty pissed off about Watson and Crick. So he's the one who discovered this Chargaff's rules. If you read the double helix from Watson, you will remember Chargaff's rules figure centrally. And here's what he says. So far as I could make out, they wanted, unencumbered by any knowledge of the chemistry involved, to fit DNA into a helix. And all I would say, again, this is for the young folks in the room, is who came out on the winning side of that one? And there are a lot of examples in the history of science of the senior citizens saying you don't know anything, and there's all this established stuff, and then there's just middle finger. Just go for it. So anyway, why do I bring it up in this context? Because the gene I'm telling you about is actually about this story, purines and pyrimidines. And so this is the before. So we did not know what was going on. That's where that sequence came from that I showed you. And this is the after, right here. And the word discovery is a big word. It's a word I don't think I would ever have used for anything I've done in my whole life. It's kind of sad. And this is not my discovery, but the students working with me produced this. Every single one of these was a blank when we arrived on the scene. And now every one of them, we can do StatMech. In other words, we can do my real job, so to speak, which I will come to in a moment. Just for those of you that are interested in computation, machine learning, and all that, until now, we've done this endeavor by hand. What do I mean by that? I mean that we look at these things and we try to make guesses about where binding sites are. It's terrible. And if you want, I can show you afterwards. We have 8,000 of these, and we're getting ready to try and do the whole genome of E. coli and the Pseudomonas, and then we'll have 80,000 of them. So we have left the realm of it's OK to do this by hand. And so we are working very hard at trying to find ways to, in an automated fashion, examine this. OK, so what have I told you thus far? First of all, again, I want to summarize what I claim the problem is. I'm very worked up about it. And I'm more than happy to entertain emails or whatever opinions you have, I want to hear them. But I've been on this rampage for 10 years, and thus far, nobody's convinced me that I'm missing the point, OK? Although a lot of people think I'm missing the point, I'm not convinced by what they have to say. It bugs me that there are 10 to the 17th letters, and yet even in E. coli, for 60% of the genes, we do not know how they're regulated. If you were to go to the bottom of the ocean in Alvin, as does, so funny, Victoria Orphan was here today. I'm about to show her picture. It was in my talk to start with. She just got back from the Aleutian Islands. She went digging around in the sediments. She grabs some soil from the bottom of the ocean and finds organisms that have never been seen before. We know nothing about how they're regulated. So what's the objective? Like, so what? Let's say we pull this off. What's the goal? What will satisfy me or you or others that we can make some forward progress? And in a way, what I want to say is I want to understand the physiology of organisms and their evolution. And I'm going to come back to whales again. If I were to go to sleep like Rip Van Winkle for 100 years, and I could come back after 100 years, and I could ask a scientific question, I would say, could I please go have dinner with whales so I could talk to them and hear about life in the ocean? That's my honest, for real thing that I would want to do. Have we learned to talk to whales yet? So I'm big on them. If anyone wants to understand more about why, I'm happy to tell you. But this is a very cool graph. On this y-axis is the maximum time of diving. On the x-axis is the mass of the animal. And amazingly, these are all fossil whales. None of them exist anymore. So people have a sense of how long they could stay underwater. Why do I bring that up in this context? Because they have myoglobins, which is shown over here, that are different than my myoglobin and yours. And there's all sorts of genes which behave differently because either of elite diving or elite flying, like the bar-headed goose, which flies over the Himalayas. All of it, in a way, comes back to this kind of question, which is, what's the input-output properties of this system, and how do we think about that? Well, I want to compute the behavior, let's call it, here I'm saying the fold change, which is the change in the level of output, as a function of some control variable. In this case, I'm thinking of pipetting in something at some concentration. And there's a very generic characteristic to such curves. They have a leakiness, which I claim is very interesting to calculate. They have a saturation. There's a midpoint, which I will refer to as EC50. And then they also have a slope. In other words, how steep are they at that midpoint? All of those are things that one would like to calculate. And what I'm going to claim, based on what I told you earlier, is that for every one of these architectures, we and others know how to use equilibrium statistical mechanics, chemical master equations, broken detailed balance. There's all sorts of different ways of calculating what these things will behave like. Forget the equations. It doesn't matter. What would you do with that? Well, you can ask super important questions, like, for example, how do stem cells end up becoming the different kinds of cells that are in our immune system? Every one of these red sets of blunt-ended arrows is what's called mutual repression. That is where I repress David and he represses me and one of us wins. And the way that that win takes place is either I'm high and he's low, or he's high and I'm low. There's two stable fixed points of that dynamical system. And so, in a way, that's the mathematization of these ideas. So I just want to foreshadow for you what we do when we know the regulatory architecture. So here's an example where it's a very simple architecture called simple repression. This was originally formulated by Jacob and Monod in the early 60s. And they called it the repressor operator model. The idea is that there's a gene. It's on unless the repressor is nearby. And what I'm showing you is using either statistical physics or using kinetic equations, we can compute level of gene expression, versus number of repressors. Let's just qualitatively understand that. The more repressors there are, the more bouncers there are at the door, the club, the fewer people get in. So that's why the x-axis has reduction in gene expression. This axis, these different curves correspond to tight binding to the DNA and weak binding to the DNA. So those are predictions. They're parameter-free. And so we can do even more sophisticated things. And this is 15 years of experimental work. And I beg you to believe me, all of these curves existed before the experiments were done because they forced us to design the experiments. And that's cool. That was a very fun endeavor, but it got way better for reasons that I'm going to show you now. So I like this notion of hidden variables, and I don't think that it's gotten enough attention in the biological setting. I'm not talking quantum mechanics. I'm talking about something simpler than that. And there's this beautiful paper from Joe Keller in which he made the following observation. So if I take a coin and I flip it up at an initial velocity v0, we know that the travel time is v0 g to the top and v0 g back down, so the total travel time is 2 v0 g. If I know the angular frequency, omega, then that time times omega tells me the number of spins. And so you can basically figure out heads or tails. And once again, if I know this curve, if I know the initial velocity and initial omega and you don't, and we're playing a game of with money, I'm going home rich and you're going home poor. And so what I wanted to show you here is this is a parameter-free. There's no fit whatsoever. And to be more insulting to ourselves, that's 15 years of effort where we thought we were tuning knobs. We thought we were changing the system. And what we came to realize is that the cell was not interested in what we thought was important. It had its own concerns. And it turns out that there's a parameter which is a combination of all sorts of things such as copy number of repressor, copy number of genes, strength of DNA binding, concentration of inducer, all of which together conspire. I guess there's not really any, is there somewhere I could write? Forget it. Which all conspire to give you the denominator in a Fermi function. And so this data collapse just drops naturally out of our analysis. OK, so I'm almost done. I just want to say that this is, to me, what the goal is with understanding genomic architectures and understanding the input-output properties. So Victoria Orphan, who was just here a few hours ago, and we've been leading trips to the Galapagos for years together and are dear friends. We've articulated this thing we call the Alvin challenge. And the idea is she will go to the ocean floor. She will grab an organism that's never been seen before by human beings. And within a week is kind of the scale of our ambition. We would actually have a complete description of every single gene at this cartoon level. There'd be associated mathematics. There'd be energy matrices associated with every such gene. So the concluding thought is, imagine what we could do if we knew the rules of writing the poetry of the genome. And I guess, to hearken back to my past in condensed matter physics, I was always intrigued by things like the Iron Age or the Bronze Age. But I also am even more intrigued by the fact that tomorrow I'm going to go to Newark, and I'm going to get on a 777. That's an airplane that was designed. The president at the time of Boeing, when they were doing that, was Alan Mulally, who then went to Ford. And that plane was designed on the computer. There were no wind tunnel tests. United Airlines took the first one. They just built it, and off they went. And the difference between the Iron Age and the Boeing 777 age is really intent. It's like really understanding the details of your system so well that you can forego experimentation. And to me, that's what it would mean to have genomic poetry. So John Wheeler made this remark once upon a time that I like so much, which was the real reason for students at universities is to teach the professors. No one is that more true for than me. I've had some time with these two guys at MBL, and I think we all find that we all learn together in those kinds of circumstances. So I just want to say again, I am deeply grateful for the chance to be here. I really, truly have had kind of a long interest in Jim Simons. I love differential geometry, point one. But also I like people who live weird lives and that are ambitiously strange. And I mean that in the best possible way. So anyway, thank you very much for having me, and happy to take any questions.
thank you very much. It's a lecture that I know Jim Simons would have loved.
Cool. Well, that's a big compliment. I hope you're really sure of that.
I'd also note that for those of you who want to do some experimental work, there are 3 clubs on 21st Street with bouncers.
Nice.
Looking at the way that many of us are dressed, I think
Repression.
repression
There might be
may
repression.
the fate.
Yeah.
But yes,
Will baldness help?
I don't know. We can do the experiment.
Hi. This is a wonderful talk. Thank you.
Thank you.
This is not directly about your conclusions, but early on you showed a graph with convergence of progenitor molecules, and you showed it was linear over a scale of 300 million years.
Yeah.
But uranium has a 50,000-year half-life, so there must be like thousands of times more uranium, and I would assume mutation rates would be, pardon me?
Uranium has hundreds of millions of half-life.
Yeah.
Oh, I'm sorry, what was it that had 50,000-year half-life? One of the isotopes, but I would think that there were more radionuclides
Well, I mean, all
earlier
I can
in
tell you
the
is like
mutation
an
rate.
interesting spin on that is that the genes in insects are more stable than than India and the Chinese continent. In other words, on the time scale at which India was crashing into China and closing the Tethys Sea, genes were stable. But I'm not sure what your question is, but maybe your point was just about the 3 different molecular clocks and things like that.
Thinking that mutation rates were higher in the past.
I see what you mean. Yeah. Yeah, I know people have thought hard actually about these questions, and I think that the question of the molecular clock is robust against that, but it's a very subtle thing. In other words, the molecular clock is it's a subtle business that I guess I don't want to. I was just trying to tell you about using molecules as documents of evolutionary history, but that's very, very tricky stuff. I don't know about the connection to higher, earlier rates.
In the back?
Hold on, I'm coming over.
I've long been intrigued by the picture that you ran in Physics Today with Stephen Quake about the convergence of energy between 10 and 100 nanometers.
Yeah.
And I know that this has inspired the current, study of mechanobiology.
Yeah.
And I was just wondering if you were still thinking along these lines.
Very much so.
Convergence of energy.
Yeah. So let me repeat the question. So Quake and I wrote an article called "The Biological Frontiers of Physics" in Physics Today, like in 2005, and we're just where this came from was pretty simple, which is when we order primers for PCR, the temperature at which you melt DNA is not very far from thermal energy. And so another way to say it is KBT is 4 piconewton nanometers. It's 4 times 10 to the minus 21 joules. And lots of biology teeters on the edge of thermal energies. So that was observation number 1, which is you're not that far from thermal energy for lots of things. Like even a virus, you heat it up, it will actually fall apart and so on. So that's point 1. And so we got to starting to wonder about other processes, like the Debye length and the Jerem length. And there's all these examples where you could say, well, what is the role of thermal energy in this thing? I would say I've got probably one main passion in science, which started at age 17, and it's exactly this point that you just raised. I can frame it in different ways. Number 1 is, what are the rules of non-equilibrium? That's the biggest sort of casting of the problem. But a different way of putting it is that, we're made of the same elements in the periodic table as stuff like this, and yet we're clearly different living organisms, and energy must be invested. You don't invest energy, and that story I told you about the fidelity of replication, gone. You get 1 in 100 error rate if you don't put in energy instead of 1 in 10 to the 10th. And I can go on and on like that. In other words, you have to invest energy or you're out of business. So anyway, I guess all I'd say is that I think it's one of the great themes of modern biology, to be perfectly honest. I think, what is it? For sure, but that's part of biology writ large. Yeah. I ran into Jim Hudspeth this morning. You know, he's a hero of mine. And, you know, if you think about hair cells, there are thermal fluctuations. And the question is, how do you actually make a measurement when you're in the face of such noise? And again, energy investment allows biology to, in a certain sense, avoid things like, I'm kidding when I say this, but like the diffraction limit. There's ways of getting around these kinds of effects. Anyway, I don't know if that was a good enough answer, but I'm happy to chat about it more.
Beautiful. So much of what's interesting about the way evolution has happened and the variance in phenotype between individuals, but also between closely related species, almost certainly has to do with relatively subtle differences in regulation. So it's very intriguing and important to do exactly what you're describing. Do you think you're ultimately going to be able to say, though, that we understand well enough to say a subtle, I understand the promoter, let's say.
Yeah.
I understand that well enough to be able to understand how subtle changes happen.
Yeah.
And really it's the impact of those, probably in aggregate,
Yeah.
genes, that's really resulting.
Yeah, I think your framing is perfect. And I guess what I would say is that, you know, the point of these energy matrices, this thing over here, and we've already gone very far down this path, is you can tell me a sequence, and I can tell you the binding energy, and hence how I'm going to change the level of gene expression. Just to give you one example, there are more than 40 species of flightless cormorant on the cormorants on the planet. One of them is flightless. It's in the Galapagos, and basically we know what's going on with the gene. It's basically a gene expression thing, and so so I think at the level of individual nucleotides and, you know, changes to repression or activation, all that, I think it's possible we'll get to some point of understanding that. But then I think there's a higher level thing, which is really, really out of my pay grade, which has to do with what you just said, which is like, how does that insinuate itself into the bigger questions? I'm sure you're well aware of all these to me, very strange contested debates where people want to say it's about the protein coding regions or the regulatory, like why do we have to have it be one or the other? I just don't really get that. I feel very persuaded that regulatory evolution is critical to different organisms, and yeah, I'd love to talk to you about it. Like what's ways to approach this that are actionable and meaningful, like where you'd be satisfied.
So I work on human papillomavirus genomes.
Yes.
8,000 base pairs. They cause cancer. You know, trying to get at the genetic basis of what is it about one genome versus another. And it's led us down the line to understand that, to try and get at what is the information content in DNA. The genome has evolved so beyond the central dogma, you know, and even the non-coding elements. How do you measure, how do you get at this, you know, information content that's, you know, I mean, I could talk to you later about, you know, kind of analytically trying to get at, you know, what is the specific nucleotides that are responsible for cancer. But so the question is, how do you get at the information content of DNA beyond, you know, what we know?
Yeah. So, so first of all, what I was telling you about is definitely a little steps for little feet thing that has to do with transcription factors. There's a really cool paper by Bonnie Bassler that's in PNAS in which they looked at regulatory RNAs. And what I'm saying is they did the same scheme of doing mutagenesis and then reading out what that implications were for, this is in the context of quorum sensing. I have no reason to doubt that such things will work in the context of viruses. In other words, I think that's a very good, this kind of, if I were going to spend money on looking at a problem, and I've worked on viruses a lot, I think it would be super fun and interesting. I agree with you, you know, they're super compact genomes, you know, like SARS-CoV-2 is big at 30,000. And they have multiplexing and all sorts of things like that that are subtle. Again, my answer in a way which you're not going to be satisfied with is little steps for little feet. You know, like until I can do E. coli, I'm going to imagine that I'm kind of stuck. But I think I would love to talk to you about, you know, whether or not you buy that mutagenesis of the kind I was describing could be useful.
So we'll take one last question. I know there's more, but we're
Hi.
Hey.
I was wondering if it's possible to parse DNA using things like formal grammars, like the Chomsky hierarchy, if you're familiar with that.
A little bit.
I mean, you have the hierarchy, regular languages, context-free, context-sensitive, and then unrestricted grammars. I would imagine it has to be something at least context-sensitive. So I don't know if there's any work in that direction.
I think there are people working on that, and we need friends like that. In other words, when I just told you we're going to have 80,000 of these information footprints, that is the kind of thing that we're after. And I don't even know if I would recognize it at this point. So, that's the beauty of people that know different things talking to each other, I guess I would say.